And we have two very eminent speakers for this evening session, Professor Tom Blundell and Professor Jane Richardson. So what I'll do, I will request Professor Saudamani to actually introduce Professor Tom Blundell to the people attending this conference here. So Professor Saudamani, please.
Yes, yeah, thank you. It's indeed, I'm very happy to introduce Professor Tom Blundell, who actually needs no introduction to this audience. He had received his DPhil in the Oxford University, where he worked with Professor Dorothy Hodgkin and had solved the crystal structure of insulin. And subsequently, he had also been very popular for a long and deep study of many structures of aspartic proteinases and related enzymes, namely the HIV proteinases. And on the protein crystallography, the book by Professor Blundell and Johnson is very well known, and it's almost like a Bible for the crystallographers. But not only on protein crystallography, but equally so on the bioinformatics as well. Professor Blundell has contributed very directly and looking at insulin and related proteins, comparing aspartic proteinases with the HIV proteinases. His group had performed or initiated several comparative modeling software such as Composer and Modeler, which are world famous. And Professor Blundell had also initiated a company which approaches drug design from a fragment-based modeling, and it has been incredibly successful. And several drugs from Aztecs has come over to the clinical trials. And apart from his scientific interests, Professor Blundell has a number of contributions as a science administrator as well. And he has served in the AFRC, and he has also been an advisor to the UK government. And from Oxford University, he had spent time in the Sussex, and then in Birkbeck College, he has served in the crystallography department for a number of years. After 1996, he moved to the University of Cambridge by accepting a William Dunn Chair professorship. He continues to work in signal transduction, and he has solved a number of structures like crystalline, pentraxin, stromalysine, and several proteins in DNA repair. Professor Blundell has been awarded many awards, and I wouldn't be able to list all of them, but some of them are like the Krebs and the Pfizer Award, and he's a fellow of many societies, including EMBO, and he's a foreign member of Indian National Science Academy. He received his knighthood in 1996. He has very strong and long association with India, almost since 1970s, and known some of our own gurus like Professor Vijayan and Balram closely for these years. Both Srinivasan and myself were fortunate to have spent time in his lab for nearly 6 years. And during this time, I found that apart from our assignment of 5-psi regions for amino acid substitution tables, he had always struck very insightful conversation with us on Ramachandran map. Indeed, he has a deep respect for Professor GNR, and I'm all excited to hear it straight from him. Professor Blundell, please.
Well, can you hear me?
Yes.
Well, thank you very much for the kind introduction. Can I call you Minnie in this?
Yes.
Others may not know that when Srinivasan and Saudamani joined our lab, I found it very difficult for my colleagues to remember those names. So they got called Minnie and Srini. And I think those names have existed for now quite a few years. So what I was going to talk about today is really not about the actual work of GNR, but really the very broad impact that his ideas have had on what I've been doing over the years. And these are displayed on this slide. So The question now is how I get it to work. Okay, so my own history is that I started to work with Dorothy Hodgkin on insulin, insulin crystals, and got interested really in the early 1960s in Dorothy Hodgkin's work. I was interested in that because not only did I know a little bit about her work, but I knew also that she was very interested in politics. I was a left-wing Labour councillor in the city of Oxford and ran all sorts of radical clubs when I was an undergraduate. And I found that Dorothy Hodgkin's husband was heavily involved in politics and very interested in Africa, which I was as well. And so I went, it's now around 60 years ago, I went to look in her lab to see what she was doing. And I spent a term there while I was an undergraduate in, I think, 1961, so 61 years ago. And I was very impressed and decided I would go back once I finished my first degree. I did very well in that. I was very lucky. And so I got a PhD studentship. And rather than join Dorothy to begin with, I thought I'd better learn the methodology. And so I worked with a colleague. And then when I finished my PhD, which I did in a couple of years, I joined Dorothy Hodgkin's group working on those insulin crystals you can see there. And so I joined Dorothy Hodgkin in 1964, but in 1967, we got the structure of the insulin hexamer. It was the first crystal structure of a protein hormone and one of the very first crystal structures of proteins. And so I was fascinated by the implications it had for understanding the storage of the insulin and thinking about its functionality. So that was a key point in my life. But as I looked at the protein structure we had in 1967, then I realized I had to understand the conformation. And I discovered that 4 years before, Ramachandran had actually published his plot that you can see there. And so I got interested in that. And of course, we had many Indians in Dorothy Hodgkin's laboratory, including some very distinguished members and also some young members like Saudamini, who have since become very impressively productive. And so that was a very nice experience. I went on to get very interested in the application of structure to function and understanding drug discovery. And I've done that in cancer. I've done it recently with the SARS-CoV-2. And then, of course, there's been a rather radical change over the last few years in that we originally looked at everything using X-rays, but for the last 10 years, we've been focusing very much on cryo-EM. And over the last 3 or 4 years, as the software methods have improved, we've been looking at these huge complexes. So in biology, you need to know not just the single components, but you need to know about the multi-protein systems that are going to be functional. And I'll tell you a little bit about this one you can see here, which is involved in DNA repair. And so you can see that it's a multi-protein system. And I could have never done this with X-rays. So there's been a big revolution. And even in my company, we knocked down half of the building to put a cryo-EM machine in it, as well as we removed almost everything, including, I'm afraid, NMR machines and so on, from the basement and put cryo-EM there. And so that's been the real revolution. And it's allowed us in the company and in academia to move into basic science and drug discovery. So just to go on a little bit about the background, of course, the great influence on my life was Dorothy Hodgkin, but I have to admit that I'd heard and read about J.G. Bernal even before I heard of Hodgkin, because as a school kid, I got interested in reading things like those that Bernal was writing about the principles of science and trying to understand it. And then I realized that Dorothy Hodgkin, when she was just 21 years old, I think, moved from Oxford to Cambridge to work with J.D. Bernal, and he became a huge influence on her life as well. And so between them, they pioneered putting crystals in a tube with the mother liquor and getting diffraction patterns for the first time. So this was in the 1930s, really, and they wrote an amazing paper in 1934 in Insulin, just predicting that now you could see things by doing X-ray diffraction. Then we can look at lots of crystalline proteins and understand structure and function. That, of course, was correct, but what wasn't correct was that you could do it quickly. So this was 1934 when that paper came out in Nature, and it was, as you can see, another 20 plus years before things started to move in Cambridge, hemoglobin, and it took us until 1967, really, to get the structure of insulin. So Dorothy was way ahead of the time, but it took decades. And that was a very important lesson for me, that if you're going to make a huge contribution like Dorothy did, you might need to concentrate on it for a long time. So that was Dorothy's thing. She took the insulin crystals, she got diffraction patterns on it, and she also looked at crystals in the pancreas and realized that actually the crystals of insulin were actually mimics of the very tiny crystals that are used in the pancreas in the islets of Langerhans to store crystals. So this was a team of Dorothy's, and you can see that Siv Ramachandran was there in 1964 when I first went into Dorothy's lab, but working with a colleague at that time. And on the left-hand side, you see people from Australia and New Zealand and UK. And then I joined in 1967, Dorothy's group, and Vijayan, that you all know, became president of your academy, joined as well. We were roughly the same age. I think he's a year older than me. And we became very close friends. We're both there when we solved the structure of insulin. But it was an international team with a lot of women in it, and a very communicative and interactive place. And you can see we had parties together, very unusual for that period of time, to have a multinational, multilingual, balanced group with women and men doing good science. That was a lesson I learned from Dorothy. We also, of course, when we solved the structure of insulin in the late 60s, I realized that it was beautiful. And I've been brought up in a family that did art, no science. My grandfather was a professional artist, and I'd done even art at school right through until I was 18, 19, and then discovered I was probably better at science. But I saw beauty in the insulin structure, which reflected in many of the artistic aspects, like the Chartres Cathedral window here. And so we began to think about beauty, about function and structure. And that's myself at the time with Dorothy in discussion. And you can see my hair was slightly longer then, and I was rather unconventional, although I was the city councillor running the city of Oxford. So that was the beginning. But it was then when we had the structure of insulin that I suddenly realized that this paper had been published by Ramachandran and Ramakrishnan and Sasisekharan, of course, on the Ramachandran plot. And in the 60s, we therefore, when we got the structure of insulin, started doing lots of analysis. And I was able to begin to understand, for example, on the right-hand side of the plot, you've got left-handed alpha helix, but you also, of course, have glycine over there as one of the residues. And I realized that the glycines in insulin had positive phi's, and they were very heavily conserved. So I began to understand from the Ramachandran plot some of the restraints on evolutionary change in proteins like insulin. And so I wrote a lot about evolution at that stage. But at the same time, I was heavily involved in politics. And I took over the city of Oxford as a local politician. I stopped a motorway coming into Oxford in 1970. I tried to get representation for people in different areas. That was where the motorway was coming with red arrows. And I wanted to conserve not only the beautiful buildings, but also the townscapes that you see in the top right-hand side. So I got heavily involved in politics. And so while I was getting papers in Nature with Dorothy, I was also running the city of Oxford as the chairman of the planning committee when we took control of Oxford. And nothing that I did there has ever been changed. We stopped the motorway going through Christchurch, Meadow, and so on. And we kept North Oxford, which had a lot of houses like this. They weren't individually nice, but the townscape was beautiful. However, I realized doing politics and science, life was rather complicated. These are things that I did while I was a city councillor. And I decided I probably had to go away and think. And so I had a great dilemma. And I decided what I would do is to take a year off and travel the world. And this is the route I took. I went, I think very, very few people in Europe when there was Soviet Russia had taken the Trans-Siberian Railway. I went all the way by rail up to the beginning of the Trans-Siberian. I went across there. I got to Siberia, got stuck. I found a Japanese lady who'd taken some people or men who'd fought in against the Russians and lost their lives. She'd taken them back when the Russians and Japanese were trying to make friends. And I met her. She had a plane organized. And so I was able to fly out of Siberia and land in Japan. And there I lived for a few months and began to learn Japanese, so Nihongo Benkyo Shimashita. And then I decided I better move on. And moved from Japan through Taiwan. I couldn't get into mainland China at that time through, as you can see, Thailand, and ended up in India. And so this was my first visit to India. I'd already, of course, had several people from India working with Dorothy, and I knew that Bangalore was the center. And so I made my way down to Bangalore. But I had met lots of friends. You can see this is a picture of Dorothy Hodgkin. And Dorothy was one of the few scientists from Europe who'd been several times to India, even in the 1950s and so on. And you'll recognize some of the faces around here as very important people. So Dorothy Hodgkin, through visiting India and having Indians coming from particularly Bangalore, but also many other places, had a very close friendship. And this allowed me to know many people in India and to get involved. And of course, one of the main people I met was Vijayan. And the other thing that I did was to do music, which I haven't got a slide on. But during this time, I also began to think about evolution and about the work that Kimura had been doing, and in a much more broad area. And then also thinking about what I was going to do. So one of the things I did was to write a textbook with Louise Johnson when I got back. And Louise, you see in the bottom here with Dorothy Hodgkin, but she was actually working with David Phillips, who became Lord Phillips in the end, but was a very influential, very thoughtful person, and very keen to have women doing science as well as men. There weren't too many at that time. And so the Phillips group and the Hodgkin group started working together, and that led me to write with Louise Johnson this first text on X-ray, really protein crystallography. So that was what I was doing, but I was also getting involved with many companies. That was rather unusual for somebody who was very left-wing. And the reason I was doing it was, of course, that insulin was produced in companies. And I found that Novo, Wellcome, Eli Lilly, many other companies were interested in what we were doing, and they could provide us with insulin to do studies. So I was able then to start thinking much more broadly about the interrelationship between basic science and applied science. So I left Oxford and I went via Sussex and ended up in Birkbeck College, where I was the successor to J.D. Bernal. So I found myself, when I was 34 years old, having traveled the world and done lots of unusual things, being head of a department in Birkbeck College, which was doing lots of crystallography. And so that's where I began to develop a lot of ideas, all based on what I'd learned with Dorothy Hodgkin, but not only doing basic science and left-wing politics, but working with companies as well. So one of the things I was doing was looking at pepsin, which we solved correctly, and found that renin, which was a much more important enzyme controlling blood pressure, looked like the picture I'm showing you here. And then I suddenly realized as I looked at this that it had evolved from two halves which were identical. So what must have happened, I surmised, was that there was a dimeric structure, and in evolution, two copies of the gene are fused so that the two halves of that structure that you can see on the screen here, this half and this half, were actually having the same fold. Now, I think you'd agree with me that they don't obviously at first sight look, they have the same fold. And it took me some time to realize they did. And therefore there must be an ancestor. And the ancestor would have looked like that. And so this was all happening in the late 70s. And I published a paper predicting there was an ancestor of this. And what happened is in the next 6 years, I found that ancestor was still around. And where was it?
Hello?
Hello? Yeah. And is there a problem? No?
Is this my school meeting?
Sorry, I think it's been a mistake.
Yeah. Okay.
Oh, yes, sorry.
Yeah. Okay. And what I realized was that HIV genome I could look at, and I could use the model that I was just showing there on the right-hand side as a target. So when the pandemic arrived with HIV, I very quickly...
Is anyone here named Amanda?
I don't know who's... Could you, whoever's talking at me, please don't. So what I'm doing now is showing you the HIV, and there I realized was the proteinase. And so when HIV started to develop in the 80s, I very quickly was able to design inhibitors of that and get companies to start working on the whole area. And so we looked at the single chain of the HIV proteins, found the protease and designed inhibitors for it. And they were inhibiting the polyprotein, which is the target protein. And that's the structure we published for the HIV protease. And then I designed new drugs. So I suddenly realized that in a very short time in the 1980s to a pandemic, I could design things, and lots of companies then got very much involved in what we were doing. But then I also realized that there was mutations occurring, and we had to fight resistance and understand how to design a drug and minimize the resistance. So I then got involved in drug discovery. And so I first had to go from the genome to the protein, and then, of course, from the protein to a ligand. And as I was very left-wing, people couldn't understand why I suddenly got involved in drug discovery. And then I realized that I had some ideas that other people didn't have about how I would explore biological space and then new molecules for new drugs. So an example of this, which I've been doing much more recently, is in non-homologous end joining. And this is something that repairs things in cancer, in cancer cells. And so if you can design inhibitors to it, you have an anti-cancer agent. So I started looking at this complex system of non-homologous end joining to see whether I could target in a cancer cell the ability of the cell to repair itself. And because it was doing it much more quickly than other cells, the drugs would hit it more. So I started looking at all the components of non-homologous end joining, synapses, end processing, and ligation. And more recently, we've looked at these hugely complex systems. And this is, of course, coincided with the revolution in cryo-EM. We could, of course, for many years get images, but it was really the software which allowed us to move through. So this huge system involving one protein, DNA PKCS, with 4,000 amino acids and several other proteins, I could define using cryo-EM before I could do any X-rays. So this was all going on in the last 10 years. And we published lots of papers in Nature and elsewhere of the various forms. This is in Molecular Cell, which is a very good journal. We were able to start thinking about targeting this. So cryo-EM suddenly took over, and almost everything we've been doing recently in drug discovery is there. But of course, in parallel, we also had to do some computational work, and we have been doing work on the human genome, and I got involved very much in mycobacteria. So first tuberculosis, but afterwards other mycobacteria. How do we extend the information on the human genome and on the genomes of pathogens? So I started thinking about this, and we did a lot of thinking about knowledge-based modeling. So the first software that I wrote for this, and I'd written a lot of software earlier on, was Composer. That was in 1987. But then I had a very good person, Andre Shelley, who's now a famous professor in California, of course. I proposed to him an idea about modeling using knowledge-based methods. And we started using writing this program modeler. I have to say that Andre was a brilliant computer programmer. So even though we had the idea together, he was very instrumental in developing the program. This program, just one paper we published, it's now actually had over 13,000 citations on one paper. There are some professors in Cambridge who have 13,000 citations on all their papers. So it was rather satisfying. And then we wrote a lot of other software allowing us to homology recognize and to build structures. So I then set about not only doing experimental work, but also providing databases, excuse me, for not only human and cancer, but also for mycobacteria. So one of them was Chopin, which is a database where we modeled from the genome the various targets, and we could then look at the druggability of the models. And there you see at the bottom, you see there's Saudamini, and on the right-hand side, there's Srinivasan, very sadly missed now, but the two of them came in and worked in my lab, but then went back, and we found we were working on similar things in terms of generating databases. So we began to collaborate again, although they left my lab and were back in Bangalore, and it was all their original work. So a very exciting collaboration. And we then got involved in things like leprosy, and my lab became the center of the American Leprosy Mission having its base in UK, when you can see from the picture that the actual leprosy is of course, in India, though not everybody knows the extent, in many places in Africa and also in South America. And I realized that leprosy was very closely related to tuberculosis, but not many people seem to know. So I spent a lot of time, and my lab has now become the center of the American Leprosy Mission's drug discovery. So we did genome analysis, protein modeling for that, and then have developed a lot of methods for not only protein modeling, but ligand binding, analysis of mutations, and so on. So we developed a lot of drug discovery with not only cancer targets, which I've done in my company, but also in mycobacteria. And you can see all along we've been using cryo-EM recently, getting very high resolution structures for very complex molecules. And Shereen Thomas, of course, is from Kerala, as you might guess, and Shikan Liang from China. And they've been developing some amazing high resolution structures using cryo-EM. So that was one revolution. Cryo-EM is a resolution revolution that's completely changed our lives. We don't use X-rays very much at all now. The other thing that's happened in parallel in my lab and elsewhere is the use of machine learning methods. And this is a guy called Aryan Jamas, who's a very, very clever computer scientist who's done his PhD with me and with Pietro Leo, and is very interactive, and developed a lot of software for modeling using machine learning. So the two revolutions that have occurred is, one, we moved from X-rays to cryo-EM, and then secondly, we moved from statistical methods to using machine learning in understanding computationally things. So there, what has changed things a lot. We've, of course, spent a lot of time then screening chemical libraries. And as many of you know, I formed with another Indian. You can see this story is all about Indians' contribution in my lab, really. Haran Jyoti, who came to England when he was quite young, 7 or 8, I think, but then ended up doing a PhD in my lab and then working with Glaxo Wellcome. We formed a company called Aztecs. I think some of you know about it. Originally just 3 or 4 people around in the lab and above the local shopping center, and using a fragment-based method where we took very tiny fragments and screened those. You have smaller libraries, but you need to have sophisticated techniques. So Haran then took the initiative, and I co-founded with him Aztecs. We had 80 employees very soon. Chris Abel, who sadly recently died, was the chemist, joined us, and we built a big lab in the science park after Haran had spent a little time in large companies like Glaxo. So that was all very exciting. And out of that commercialization, we've got a large number of drugs in clinical trials, and we sold the company. So for a left-wing scientist to suddenly sell a company for nearly $1 billion was a bit of a surprise to everybody. But the company's been very successful under Haran, so it's an Indian initiative, really, in developing these fragment-based methods where we use very small fragments to get drugs. It's almost every company in the world is now using similar techniques. So the most successful drug we had for advanced breast cancer that's now selling about $1 billion a year. This is ribociclib. We developed it with Novartis. You can't take things through clinical trials if you've got a very small company, but it was the drug we invented. And then we've done other things for metastatic urethral carcinomas and so on. So out of the academic left-wing background, we had a company, and then we started to use that company's ideas for areas where you can't really do anything in companies. So in tuberculosis, leprosy, and cystic fibrosis. And so we, of course, got very much involved in Southern Africa. And Bill Gates I met, and he has put a lot of money into this initiative, and we've helped him spread from tuberculosis and develop using the techniques we've been developing on tuberculosis, using these fragment screening methods. You see in every slide that I've got, there's Indian, there's Sachin in this slide, and at the bottom, Pooja, both Indians who came and joined my lab, using biophysical techniques to screen fragments and to get new drugs. So this is all the fragments that we look at using X-ray or cryo-EM, and we then put those together and make new drug-like molecules from the very small fragments. And this is Bill Gates joining us. We made him put a lab coat on and having a big discussion about the methodology there. So Bill Gates was very interested in the methods we've developed, and we had a lot of very good collaborations with him. I think it was very, very exciting. He's a very original thinker, as well as a very rich person. So that was very nice. And then, of course, we had to face drug resistance. And so what we've done over the last years is to develop a lot of software for really countering drug resistance. We've used statistical methods. I'm not going to go through these, but you see that as usual, there's Aaron Prasad here, Indian scientist that I recruited when on one of my trips to India, who's developed some amazing statistical potential energy functions and is back again working with us at the moment. And then we've also used machine learning to predict the effects of drug resistance mutations. And this has been done mainly by a guy called Douglas Pires, who is from Brazil, but has now moved over to work with David Asher, who's another person from my lab in Australia. So this is software for understanding drug resistance. And of course, one of the focuses or the foci of our work is to try to design drugs in a way where resistance is less likely to occur. I think that's been very important in studies we've done. So we've been through all of these processes. We've been thinking about how to design drugs, and if you get resistance through repurposing, fragmentation, regrowth, and various other methods. And that's the end of my story, but you can see that my team is rather unusual for an English team. There is one or two English people here. Amanda Chaplin, who's just gone and got a professorship, is English. Just going across here, not too many other English. There are one or two down the bottom here, but you can see that Indians like Sherin Thomas and Sandeep Chaitanya, who's the leprosy chair, International Leprosy Mission, Sheikh Arif, and many others who've Pooja Gupta, I mustn't forget, but also people from all over the world in our lab. So it's been very unusual as far as labs are concerned, which are usually much more English-based. But we have the advantage of the English language, so that's much more used, so we get lots of people. And then I have to pay my respects to Dr. Sibanda, whom I married over 40 years ago, and she's done extremely well. And our daughters have both been through local schools and are doing brilliant things. So that's my story. The messages are that science must be international, interdisciplinary, and gender balanced. And you can see the evidence. My current team is that. You can see from the list at the bottom, we've got mathematicians, physicists, engineers, computer scientists and chemists, biochemists and medics all working together in the lab. And so that's my story. I've taken rather a long time, but you can see that GNR played a major role in a key place right in the center of the development of our ideas. The Ramachandran plot has played a big role, but you can also see that many Indian scientists have contributed, including Saudamili, to many areas of the work that we've done in the UK. Very few English, it's an international team. So I have to thank you for being patient and listening to me. I'm happy to answer questions if you have any. Thank you very much. Minnie, it's all yours.
Yeah, thank you, Tom. I understand from the main coordinator that this being a celebration, we shall not have our regular answer sessions. So I'll wait for one of the visits when I can ask some questions. Many of us will have more questions to ask you at that time.
Yes. Well, I'm very happy to contribute. If anybody has questions, they can email me. You can find my email address very easily. And we're moving from the university up to the hospital this year. So though I'm a little bit older than most people doing science, I still have a lab if anybody wants to come and join. But thank you, Minnie, for your contributions.
Thank you for a wonderful lecture, Tom.
Yes, I think you've heard it before. We made some progress in the last 1 or 2 years. Okay, so I leave you, and you, anybody who wants to talk with me is very welcome to contact me. Thank you very much.
Thank you, Professor Blundell.
Yeah.
Thank you.
Will somebody close me down, or I just leave, Joy? Yeah. Okay, thank you very much.
Yeah.
Is that right? I leave? Yeah. Yep.
Yeah, you can just shut your presentation and yeah.
Yeah. I can't let it. Okay.
And also you have to unshare your, stop screen sharing.
I leave my share, yes. Okay. Well, goodbye, everybody, and thank you for listening.
Thank you.
Thank you.
So, Professor Saudamini, will you continue?
Yes, yeah, I'll be happy to do so. This time we have yet another star in protein structures. I'm very happy to introduce Professor Jane Richardson. Her protein cartoon diagrams are world famous. The genesis of the same happens to relate to and was influenced by her broad-based interest in various areas, such as astronomy, mathematics, physics, botany, and philosophy. After receiving a bachelor's in philosophy and physics degree, she had learned on plant taxonomy and evolution and had received a master's degree in Harvard. She then moved to the MIT, where she met her husband, David Richardson, who was solving the structure of staphylococcal nucleus. There, she started working alongside her husband, where she used her previous training on taxonomy and evolution to abstract the large structures of protein biomolecules in the form of cartoon diagrams. The cartoon diagrams have been instrumental in understanding protein structural hierarchy and also to appreciate the variety of protein structural folds. Their review on taxonomy of protein structures is one of the most well-read. Her protein cartoon diagrams are so graceful and easy to appreciate that they are universally accepted, and most protein graphic softwares, including their mole probability, have now adopted this technique into their coding. She has since contributed to several other areas such as protein structure prediction, design, and engineering. She has also won several prestigious awards such as the MacArthur Award and the Alexander Hollander Award, and she had also served as a president of the Biophysical Society. She continues to maintain a group with her husband, currently in Duke University. May I request Professor Jane Richardson to give her talk, please?
Hello, and thank you very much. I'm delighted to be at this celebration. I've enjoyed all of the meetings that I've come to in India and meeting new people, and have had Indians in our lab, but not nearly to the extent of Tom. So I'm going to do a survey that really is centered on the Ramachandran plot. And let's see, I need to share my screen. You see that?
Yes.
Great. So I've got this story about how the Ramachandran plot is alive, both in its original form. It's the most recognized validation metric of all of them for models, and relied on by everybody, and as part of the PDB validations that all the structures go through. Recently, it's been seriously misused, which is also, I guess, a sign of importance. We and other people are working on ways to get around, still checking out confirmations and getting them right in spite of this. And then it's also in the process of growing into new dimensions in a number of ways, and from other labs. So of course, at the beginning, I'm sure everybody's seen this. Normally, we just change to our modern way of doing it, but in the original version, the coordinate system was different. So the beta sheet is down on the lower left and the helix up above it, and rather than this form that we're more used to, which came from that group in 1968. And this shows a lot of the details in and in between the different main structures. Less common things on the right-hand side with positive phi and negative psi. They're not common, but they can be important locally. And then there've only been modest changes in the outlines. The main, most influential change in a way is on this left-hand side of alpha. This is a confirmation that looks very favorable in almost any of the computational procedures, but is actually quite rare in the structure. And I will mention that again later. We think we know why, and we'll go on and look at some more of these. So in the early 90s, finally, crystallography had accumulated enough protein structures that it was possible to pull out a Ramachandran diagram statistically from the known structures. This is pretty bad statistics at that point, because there were so few structures still on a statistical level that you had to include all of them. So this is from ProCheck in 1992, and it was a pretty difficult job pulling out good contours from this very noisy data, but that was enormously influential. And you can see it really doesn't have that strip of data going to the left of alpha. But in general, it just matches that original pure theoretical calculation with hand crank calculators like the one that Dave and I used in our first physics lab at Swarthmore. And then people continued to refine it. The yellow and orange contours are from the original project, which deliberately went out a long way and actually goes out into quite impossible regions, because this sort of diagonal line between the way highest point, I mean, this is log scale higher than anything else of alpha helix, it goes to basically impossible in a very short time. It's a really steep cliff. And so that's one of the mathematical problems of getting a very detailed quantitative Ramachandran measure. And then the black lines are what check, where they got around the problem of the steep cliff by only actually analyzing the very most favorable conformations. And then in Mulprobity, we've got something that really matches very well to ProCheck, which is the 98% or the 2% contour, where 98% of the data is inside that contour and only 2% outside. But now with much more data, we can make an outer contour that has very, very little. It's 99.95% is inside it, but it's very tight now because we really have enough data to do a good job on the statistics. And so this is a central part of our Mulprobity validation. What got us into this, Ramachandran was being done quite well anyway, and sidechain rotamers in a similar sort of idea. And of course, you have to get the basic covalent geometry right. What we had provided as a new tool that took us into validation, we developed it mainly for our own work, because we've always been fascinated by how proteins and nucleic acids can actually fold into these marvelously specific 3D conformations, where just to the eye, it's hard to see why one particular beta strand goes with another one. That's now, at least, there are good ways to do it. Sometimes we don't understand it at the scientific level, which I find very frustrating. I love AlphaFold, and we're having a ball figuring out what it's doing, and you know, it's an enormous aid to structure determination. But I want to know what the machine has learned that I don't know. Obviously, it has. So we're trying to find out more about it. But anyway, we developed all atom contacts by realizing that in our early protein design efforts, we had left out the hydrogens, and they're half the atoms, and they are the main thing in atom-atom contacts. So both the contact in the protein folding with itself, and also in the interactions between the protein and a ligand or another protein or a nucleic acid. And so we made a quite geometrical version of looking at those contacts. And that includes hydrogen bonds, van der Waals contacts, and then what we call clashes, when the atoms are really too close. We don't cut it off right at the van der Waals spheres, but at 4/10 of an angstrom overlap, because our analysis is not perfectly accurate to 1/10 of an angstrom, pretty good at 4/10. And atoms aren't really quite spherical, and lots of uncertainties. But when you get to 0.4 angstrom overlap, it's probably about enough to blow the protein up. So in order to make a validation set using all atom contacts, we had to redevelop things like the Ramachandran criteria in ways that matched with our contacts and could be used together. So the all atom contacts work by generating dot spheres at the van der Waals surface of all the atoms, and then looking at their touching and interpenetrations. And so we do that by taking a sphere that's only a 1/4 angstrom in diameter and running it on the surface at each dot and looking to see whether it touches another atom that's not within 3 covalent bonds. And so these are distant contacts. And when they get then to a 1/2 angstrom apart, we start putting down little dots, blue ones. And then as it gets closer to ideal, they're green. And then, the ball rolls out the other side. So we get these pairs of surfaces where the atoms are really touching or nearly touching each other. And then there are 2 kinds of overlaps. One is between donor and acceptor in a hydrogen bond, and those end up with the van der Waals surfaces intersecting. And a good H bond really is sort of a pillow, and there's quite a lot of overlap. And those that are H bonds, where there really are between a donor and an acceptor, we make this nice restful pale green. Then in other places where it's not a donor-acceptor relationship, the small clashes that we don't worry about much are in yellow, small overlaps like this one up here. And then as they get past 0.4 angstrom overlap, they have these red spikes, hot pink spikes, which we deliberately made to look uncomfortable. And indeed, people seem to find them that way and try, sometimes even a little too hard, to get rid of all of them. But basically, there has to be something wrong. One or the other atom is in the wrong place, or sometimes the name is wrong and the program doesn't recognize it. And sometimes small clashes are genuine positions. And so there can be the rest of the protein can push things together somewhat more than what you really see. But typically, in a really good structure, you don't have clashes. So both originally and then later with the X-ray validation task force, we've made later versions as more and more data was available, and also that we learned more about quality filtering our reference data. We've redone Ramachandran plots. So here at the upper left is the general case. That's 16 of the amino acids where their contours, if you do them individually, their contours are right on top of each other. And then there are a number of cases that are quite different, actually. And glycine is the most, glycine and proline are the really obvious ones that have been used for a long time, where the energetics is symmetrical. Glycine is is not chiral. And so what is possible is the same on both sides. But in fact, the data is very different because glycine is fine over on the L-alpha region. And so it's used a lot over here. On the other side, it looks quite like other Ramachandran plots of other residues, but it's quite asymmetrical and interesting, little pieces of specific things that glycine do better. Then proline, of course, because of the ring, is very limited in phi, and it's slightly different in a cis-proline. There aren't as many of them, of course. It's a little more diffuse, and it actually moves to the left in phi. And cis-proline does not have the little bump in the middle. And then Andy Karplus a while ago realized that pre-prolines were very different. Basically, the preceding amino acid interacts with the ring and makes it eats out this edge along here. That's a clash, but then right next to that edge is a favored region because there it's making van der Waals contacts. And so you get these steep edges where if you go a little too far, you're in a clash, but if you back off a little bit, you get a very nice contact. And then later we separated out Ilu and Zale, which actually are quite different. Their overall contour on the left side is not too different, but the inner preferences are quite different. And there we do get the stripe across, and it's even more striking in threonine. And I'll show you what that's doing later. And the branched C betas have a very hard time doing the left-handed alpha or any positive phi values, because since they have two arms that are really close to the backbone, they end up clashing with the backbone. So this is showing you the overlay of the contours for the 16 amino acids that go into the general plot that we're using now. And this little blip is probably threonine. We may separate it out possibly. And then over here are the other ones. And you can see how different they are. So this is actually not our current data set. We now have one about twice as big, but this is color coded on a log scale to show how many points are in these different regions and how absolutely empty the rest of the plot is. These things can be genuine. Outliers are done statistically, and so they will always have outliers in genuine good structures. In this case, it's a very tiny percent because we have so much data that we can get the contours to behave down to a very low contour level that excludes almost all the data. On almost all the other criteria, we're more at a 1% or 0.5% or maybe 1/3% level that you can exclude outliers. So none of them are as clean as this general Ramachandran plot, but sort of like AlphaFold needed the whole PDB or maybe at least 1/10 of it, according to John Jumper. He thinks he could have used 10-fold less data. And we're using the best parts of all of this data. I'm very interested to see whether if something like AlphaFold only used the good parts of the data and didn't go to 9 angstrom resolution and high B factors and invisible map density, where basically those conformations are being invented by the user in the software. that it might do even better on predicting. But it'll take us a while to figure that out and really redo the training. So, Ramachandran has been there all along. Here are the slider plots that the validation task force put together with Ramachandran there right in the middle. And everybody knows about this. Everybody uses it and tries to get rid of the outliers. And that works beautifully at reasonable old level X-ray resolutions around 2 angstrom. Anywhere 2.5 angstroms and better, it really works. And it's, as I say, it's really used. And so our overall molprobity validation really works well up to about 2.5 angstrom, where if you have good scores on everything and you also have a good match to the density, you basically have the right structure, even in the details. And that's been really marvelous for structure. However, it's not true at 3 angstrom or worse. And the reason this happens is that you can't see the carbonyl groups sticking out of the peptides. And so at 3 angstroms, the density is really smooth past that. You cannot tell which direction the carbonyl is oriented. And that's your main signal for backbone conformation. So somewhere between 2.5 and 4 angstroms, and it's not completely uniform, of course, you lose those carbonyls. And at that point, you really don't have the data in the density to orient the peptide correctly. And so you start having a lot of Ramachandran outliers in the initial fitting. And what people have been doing is then refining away the Ramachandran outliers. And so all of the ones throughout the outlier region get moved into the nearest local minimum. And unfortunately, that's not the right answer, because if you have the peptide misoriented by a lot, which happens very, very frequently, then you go to the wrong local minimum. So here's an easy to see example. This is a very nice alpha helix, not quite such good density in here. And it initially got fit somewhat wrong, probably, and then got moved into the beta extended local minimum when it should really have been in the alpha helical one. And so there's no Ramachandran outlier here. There is an outlier, a real outlier, and then a disfavored one in purple of a new validation that, well, it isn't so new now, but anyway, it's called Kablam, that looks at 5 C alphas in a row and at the carbonyl orientations to see whether the carbonyl orientations are compatible with what the C alpha trace is showing you. And those do flag this kind of error. So we've been pushing people to use those, and they've been shown very effective in the cryo-EM challenge series of tests. And what is happening, if you refine Ramachandran in a simplistic way, you get very weird Ramachandran plots. So this is from one of the earlier cryo-EM challenges where we would put together all the models from a given lab that had contributed to the challenge, and they look quite different from each other. This one clearly did not refine the Ramachandran. And that's certainly a problem because you have all these outliers and the whole plot is, very noisy. But then when you do refine them, you get strange things like these very tight plots. And again, with lines of residue values that have been pushed into the local minimum. And they also get a lot in this little left-hand region sometimes. And then this one has this vertical stop rather than a diagonal stop in alpha and beta. And again, an outline right where the refinement pushed them over a given value that is there. What they're restraining to. Now, more recently, there have been some much better statistical analyses of what a Ramachandran distribution should look like. And so in recent years, you can't tell whether they have been refined into the wrong local minimum because what they have restrained is the overall complex distribution, not just pushing things over a threshold of outlier. And so it doesn't solve the problem, but it makes it harder to see that it's happened. And so here's just a single simple case where that's the one that we looked at in the helix, where the correct answer is that it's really good helix right in very deep local minimum. But in fact, the initial model probably had it like over here or something. And so it was closer to the extended beta local minimum than it was to alpha, because the peptide was all the way around backwards almost. And so it went to the wrong place. And it really should have been here. And this is a huge difference. I mean, the conformation is just really, really different. And the Ramachandran values are wrong for both residues around that particular carbonyl. But they're not outliers because they've been refined not to be outliers. So this is a real problem. And so it's an overly tight application of the Ramachandran statistics in the wrong way. Single minimum things like a covalent angle, it doesn't hurt that much to tighten it up in order to make the refinement work better. But these multi-minimum things, particularly Ramachandran, but the same thing happens in rotamers. Putting really tight restraints on them before you have fixed the major problems, like a totally backwards peptide or a rotamer that's over in the wrong place. You really need to try to correct those things before you do this kind of refinement. Once you have corrected most of the local errors, then refining Ramachandran is great, because by then you're pretty near at least to the right minimum. Another example is, again, from the cryo-EM challenge, where this was a GROEL structure and at very low resolution in this part of the map. And you can see there's some suggestion of beta structure, but this is really very difficult density to fit into. And the Kablam outliers are showing places where there are problems. And then if you compare it to a higher resolution structure, you see these places where a peptide has been basically flipped over, but it isn't necessarily 180 degrees. It can be 100 or 90, even 60. You get a big difference. And again, of those 4 flipped peptides, 8 of the Ramachandran values are in the wrong local minimum. this one actually was close enough, and it really should have been an alpha, and so that one was fine. But the other 7 go, to different local minima. So what we're pushing people to do is identify secondary structure early, and the most common misfitting at 3 to 4 angstroms is a misoriented peptide. And you use tools like Kablam, EM Ringer, and now even AlphaFold models to try and get things into the right part of the plot. And then you can refine them, or you can just refine the hydrogen bonds, restrain them. And well, basically what Kablam does is look at longer space along the sequence. And also one can combine the phi-psi Ramachandran parameters with at least chi1 or even whole rotamers and get a much more nuanced and informative set of parameters. And so if we do another set of Rama and rotamers, we'll do them together rather than separately. So here's a neat example. Threonine is one of the most obvious places where you can see this being really helpful. This is the threonine Ramachandran plot done separately for chi1 plus trans and minus chi1, and they're very different from each other. And all of them are significantly different from the overall general Ramachandran plot. So, here in the extended region, you get nothing up top at the minus, and a big up top and not much in the bottom for trans, and then really broad, again, high, and a very big region around alpha and 310. And this one has this extra piece down here. This one goes more to the left. So if you look, and I have stuff in my way to see this, but I think this one has, oh, here we go. Yes, and trans, there's an unusual concentration here. And if you go and look at what these things are doing, this is what's called a pseudoturn. It's where the threonine is mimicking the backbone and making a hydrogen bond. So if you don't separate out backbone and side chain, there's a set of atoms that look just like a tight turn where this would be contributed by the backbone. And that's very common with threonine. It also can happen with serine. It happens with asparagine's, but with a longer turn. And so that only happens in the T rotamer, because it has to be the right rotamer to go back and make that hydrogen bond. So you can't do a pseudo turn with a minus or a plus chi1, but so all of these examples are trans, and so that makes a different shape in the Ramachandran plot. And so if you see this kind of interaction, you know that you have to make it a trans chi1, and it also has to have extended backbone. And so then the minus chi1 has this extension to the left of alpha. And we've finally seen what these do. it's unfavorable because the two successive NHs on the two successive peptides point toward each other and are actually quite occluded by the C alpha, C beta next to it. And so they can only really go to one oxygen or one water rather than having each NH be able to make a separate and better geometry hydrogen bond. And so I think it's that hydrogen bonding that means that if you were just analyzing a dipeptide, you wouldn't see this problem if you're not looking at its interactions. And so if you're looking at an isolated dipeptide, this looks like a very favorable conformation. But if you're looking at it in context and in solvent or in interactions in the protein, it's actually somewhat unfavorable. But here, this is in a helix, you can get a turn of pi helix. So there's one extra residue in the turn, and threonine, and to some extent valine ILU, where we saw also that they had points in that part of the Ramachandran plot as well. They can make this reasonably favorable by getting into this conformation where both of those NHs go back to a preceding carbonyl. And then another one of the unusual things in threonine plus, now this is the minus, that's the plus, is this little bump down here that doesn't occur in much of anything else. And that is another type of wide helix turn that does it in a different way. What happens in here is that the oxygen on the threonine inserts itself in between what would normally be the helical hydrogen bond of the carbonyl to the following NH. And so you get two hydrogen bonds, but that's still a stabilizing effect on one of these widened helix turns. So you learn all sorts of interesting things, and you get more details and little favorable spots for motifs that happen if you combine phi psi with chi1. So another case of this is looking at asparagine versus glutamine. They look an awful lot alike. They have the amide at the end, and it's just one extra carbon-carbon bond, but they behave really, really differently. Asparagine and glutamine are simply nothing like each other as a sequence replacement. They're really not similar. And so if we look at a three-dimensional plot, so here you're looking sort of diagonally into the 3D plot for chi1 trans on asparagine. It's very complicated. You know, this sort of goes at an angle, and you have particular spots that are most favorable. Glutamine is pretty well just a smear. It's Ramachandran plot is the one that's most like the average Ramachandran plot. It does all sorts of things, but none of them that well and specifically. And for instance, if you look at asparagine, again, the Ramachandran plot for chi1 trans and the Ramachandran plot for chi1 minus are quite different. You know, here you have particular values for the chi1t and the unusual spots. There are all sorts of complicated things going on here. These two, for instance, are the two ways that asparagine can interact backwards in the sequence on a helix. either the asparagine can go and make a second hydrogen bond, and that's this one, I think. And the other little plot here is that it doesn't go all the way in to make a hydrogen bond. It just sits on the outside of the helix. And these do have a minimum in between them. They're separate, where you get a clash if you try to be halfway in between. So knowing this extra detail, I think, can help us do a better job of fitting things. And we have enough data now to do it this way. So our lab is pretty small at this point. It almost always has been. We like doing stuff ourselves even more than helping other people do it. But we've had some very talented people who've worked alongside us, but in a small setting. So we also work in the middle of a big project of the Phoenix system for structural biology, and that's been a great experience. So thank you very much, and I hope you will continue to watch for development of the Ramachandran plot. Thank you.
Thanks very much, Jane. It was a splendid talk, and there's a lot of intricate details about the phi-psi values and the outliers. We couldn't agree more, and it was very important to discuss this. Thank you.
And this is the kind of place I like to be in.
It looks nice. So,
yeah.
Thank you, Professor Richardson. Thank you very much and we're sorry for the delay in getting your talk started. It was just one of
no problem for me, but I know it's getting late for a lot of you.
Yeah, it has been somewhat of a long day. So thank you very much. And at this point, I guess we have finished with this symposium. And I would like to thank all the speakers who kindly consented to, to participate and, literally educate us on a lot of the newer developments and, the nuances, especially Professor Richardson's talk on how to actually evaluate structures and to look for inconsistencies that, that are really important. I would also like to thank all my colleagues in the department for helping me organize this function. And a special thanks goes to a few of the students, especially Sri Teja, who helped in designing the poster. And then he, along with Krishnakant and Satya, for actually helping with the logistics for, the online talks and for broadcasting the talks and things like that. The members of the department, the faculty, and the staff members. So with that, we will conclude. I thank everybody and good night. Thank you very much. Thank you, Saudamini. Thank you so much.
No, not at all.
Yeah, thanks.
Thank you.
Yeah.
Thanks for giving me the chance to introduce.
Yeah, no, it was fun. I think the last talk was really interesting.
Yes.
Along with, of course, the other talks that we heard. There's so much to look out for when we're calculating structures, actually.
Correct.
Thanks, Siddharth and Saudamini and all the teams.
Thank you.
Yeah, whole day I spent listening every talk, and so exciting talk. Murthy's talk was something coming from off, and I didn't expect Anil Kumar giving such a fantastic talk. So I must thank you all for organizing. Last two speakers, of course, are there. If Professor Anil Kumar is there, I really hats off.
Thank you very much.
Oh, yes. Thanks a lot. Wonderful talk.
Yes.
Brought back a lot of nice memories and works. Yes.
Yes.
So it's a nice opportunity to meet everybody.
That's right.
Thanks to Siddharth that he got everybody. So, yes, sir. So, thank you. Yeah. I saw Ishwaran's name, but was not possible to get. Siddharth, is it?
No, he was indisposed.